Good afternoon one and all. So far from this chapter we have dealt with the introduction of cell and cell theory, and then we have looked into prokaryotic cell and then plant cell and animal cell in detail, differences between prokaryotic cell and eukaryotic cell, plant cell and animal cell. Today, and we have also done with the cell wall structure in detail. Today we are going to look into cell membrane, and we are getting started with single membrane bound organelles, of which we will be dealing two organelles today, endoplasmic reticulum and Golgi bodies. So cell membrane, which is also called as plasma membrane, selectively permeable membrane, or semi-permeable membrane. It is also called as plasma membrane because it is the limiting boundary of the plasma which is present inside the cell. Selectively permeable membrane because it selectively regulates the entry and exit of the molecules and ions in and out of the cell. It is called as semi-permeable membrane because it will regulate the movement of solvent molecules. It will allow the movement of solvent molecules freely inside the cell. Okay? It allows the movement of solvent molecules freely inside the cell. That is why it is also called as semi-permeable membrane. The clear structure of cell membrane is started to be learnt or known after the discovery of electron microscope in the year 1950. As such, many cell membrane models have been given. Of them, fluid mosaic model, which is given by Singer and Nicholson in the year 1972, is widely accepted model. Okay? Fluid mosaic model by Singer and Nicholson. In the year 1972 is the widely accepted model. So in this model, it is clearly explained that the components of cell membrane includes, what are the components of cell membrane? These include proteins, lipids and carbohydrates. These are the components of a lipid bilayer. Proteins include three types of protein molecules. They can be peripheral proteins, transmembrane proteins and integral proteins. Fine. Lipids have a hydrophilic head and a hydrophobic tail. Hydrophilic polar head. Okay. Hydrophilic polar head. Hydrophilic polar head and hydrophobic non-polar tail. Hydrophobic non-polar tail. Fine. And then if you see carbohydrates are present in the form of glycolipids and glycoproteins. Okay. They are present in the form of glycolipids and glycoproteins. Fine, right? So this is the structural composition or the components which are present in the cell membrane. Next, so here if you see, from the integral proteins, peripheral proteins and transmembrane proteins, where are they present? You look into the diagram properly. Peripheral proteins are present either on the either ends, either towards this end or towards the lower end. Okay. So peripheral proteins are present on the either of the ends. Transmembrane protein channels, they will transpan two sides of the membrane. Okay, any of the side is transparent. This is a transmembrane protein. This is a transmembrane protein channel where it transpands either of the sides of the membrane. Integral protein is embedded inside the membrane. Integral protein will transpan two sides of the membrane. Integral protein is present, integral protein channel is present inside the membrane. Excuse me. PGT is postgraduate teacher entrance exams. Okay. About PGT and its examination, in this playlist first class only I have done, you can go through it. So here if you see peripheral proteins, transmembrane proteins and integral proteins. So here you confirm that peripheral proteins are what? They are present on the either sides of the plasma membrane. Transmembrane protein channel will transpan any of the sides or both the sides. Integral proteins are embedded inside the lipid bilayer. So here if you can observe, all the integral proteins can be transmembrane proteins, but not all transmembrane proteins can be integral proteins. A very important statement to be noted. Okay, all integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Fine. And then when it comes to lipid bilayer, so most of the composition is made up of lipids only. Most of the composition is with proteins and after that it is lipids. Quasi-fluid nature of the lipids will allow the lateral movement of proteins. Here the quasi-fluid nature of the lipid bilayer will allow the lateral movement of the proteins. Okay. So fluid mosaic model is elucidated from the cell membrane of a RBC, okay, red blood cell, wherein it has this erythrocyte is having 52% of protein content in it and 48% of lipid content in it. Okay, 52% of protein and 48% of lipid. Right, understood? So here if you can see lipids, the quasi-fluid nature of this lipid will allow the lateral movement of proteins, and carbohydrates are just 5 to 8%. They are existing in the form of glycolipids and glycoproteins, and these carbohydrates which are present in the cell membrane will aid in the function of cell to cell interaction. Okay, they will aid in the function of cell to cell interaction. Fine. And then next if you see the functions of plasma membrane. Functions of plasma membrane includes cell growth, formation of intercellular junctions, secretion, endocytosis and cell division. These are the major functions of plasma membrane. Now we look into the major function, that is transport. Okay, transport in plasma membrane can be classified into two modes of transport. Okay, one is passive transport and the second will be active transport. Okay, passive transport and active transport. Passive transport includes passive transport, active transport, and there is one more type of transport called as bulk transport. Yeah, bulk transport. Active and passive transport. So passive transport can be in three modes: by simple diffusion, facilitated diffusion, facilitated diffusion and osmosis. What is passive transport? Passive transport is nothing but movement of the molecules, be it liquids or gases, from high concentration to low concentration without the requirement of any energy, without the requirement of ATP, movement or transport of molecules from high concentration to low concentration is called as passive transport. Simple diffusion is nothing but simple diffusion is just transport of gaseous molecules from high concentration to low concentration without any semi-permeable membrane. Facilitated diffusion is nothing but transport of molecules across the concentration gradient, that is from high concentration to low concentration, through the help of either channel proteins or carrier proteins Fine. Through the help of either channel proteins or carrier proteins, the transport of molecules across the concentration gradient from high to low is called as facilitated diffusion. Okay. Mostly neutral solutes will transport gaseous molecules, and neutral solutes will transport across the simple diffusion. Ions and molecules, they transport across the facilitated diffusion, and osmosis, transport of water molecules is mediated across what? Osmosis. Transport of water molecules is through osmosis, that is transport of molecules from high concentration to low concentration through the semi-permeable membrane, or you can say movement of molecules across the concentration gradient through the semi-permeable membrane is called as what? Osmosis. These are three modes of passive transport. Active transport is also of two types. One is primary active transport. Second will be secondary active transport. Okay. Primary active transport is nothing but movement of molecules down the concentration gradient, that is from low concentration to high concentration through the semi-permeable membrane by the gain of energy, where the ATP energy is required for the movement of molecules from low concentration to high concentration. This is called as active transport. Mostly the ion channels, for example sodium potassium pump, these are being activated through what? Active transport. Next is bulk transport. Bulk transport is of two types. One is endocytosis. Okay, endocytosis, and second one is exocytosis. Endocytosis is again of two types, pinocytosis or phagocytosis. Endocytosis is again of two types, pinocytosis or phagocytosis. Okay. Bulk transport is of two modes, endocytosis and exocytosis. Endocytosis is entry of molecules in bulk into the cell. Okay. Pinocytosis is also called as cell drinking. Wherein this is the entry of water molecules or solvent molecules in bulk. Okay. Phagocytosis is gaining of food molecules or entry of food molecules in bulk into the cell is phagocytosis. This is also known as cell eating. Okay. Phagocytosis is also known as cell eating. Fine, right? And exocytosis. Exocytosis is also known as cell vomiting or ephagy. Cell vomiting or ephagy. Fine. This is all about the modes of transport that are being observed inside the plasma membrane. Okay. First is what? Passive transport. It can be in three modes: simple diffusion, facilitated diffusion and osmosis. Then active transport, primary active transport and secondary active transport. Bulk transport can be of two modes, endocytosis and exocytosis. Endocytosis again based on the transport of molecules, it is of two types, pinocytosis and phagocytosis. Pinocytosis is cell drinking, phagocytosis is cell eating. Exocytosis is what ma? Cell vomiting, which is also known as ephagy. And then if you see endomembrane system first when we have to learn about single membrane bound organelles, first we have to finish off the those organelles which are included in endomembrane system. Endomembrane system includes those organelles which are endoplasmic reticulum, can be rough endoplasmic reticulum and smooth endoplasmic reticulum, and Golgi bodies, fine. And then third organelle will be lysosomes and vacuoles. These are the organelles which are included under endomembrane system. All the functions of these organelles, are coordinated. Functions are coordinated. So after completing these organelles, then we will be going into microbodies, peroxisomes, glyoxisomes and other single membrane bound organelles that are being observed both in plant cell and animal cell. We will also look into cytoskeleton, proteinaceous filaments, microtubules, microfilaments and intermediary filaments. So here first if we have to look into endoplasmic reticulum and Golgi bodies, these two organelles we will cover up today. Okay. First, in this endomembrane system, if you have to see endoplasmic reticulum are network or reticulum of tubular extensions from the nuclear envelope. What is endoplasmic reticulum? Network or reticulum of tubular extensions from the nuclear envelope. So here if you see, so this reticulum or network of extensions can be of two types based on the presence or absence of the ribosomes. So here if you have to observe, the endoplasmic reticulum, those which are having the ribosomal structures. Ribosome has two subunits, right? Ribosome has two subunits, larger subunit and smaller subunit. Larger subunit is 60S, smaller subunit is 40S. So what type of ribosomes are present in a eukaryotic cell over endoplasmic reticulum? 80S type of ribosomes. Okay. So here the larger subunit of this ribosome is attached to the endoplasmic reticulum through the protein connection, that is ribofourin. Remember. Okay. With the help of this ribofourin, the larger subunit of the ribosome is connected to the endoplasmic reticulum. When ribosomes are present over the endoplasmic reticulum, this type of endoplasmic reticulum is called as rough endoplasmic reticulum, which is purely involved in protein synthesis. This is purely involved in protein synthesis. And smooth endoplasmic reticulum is involved in lipid synthesis. Smooth endoplasmic reticulum in lipid synthesis. If it is an animal cell, along with the synthesis of lipids, smooth endoplasmic reticulum will also synthesize steroids. Okay, it will also synthesize steroids. Along with that, it will regulate the muscle contraction by uptake and release of calcium ions. It will also regulate the muscle contraction by uptake and release of calcium ions. Fine, right? So this is the complete structural and functional description of the endoplasmic reticulum. Now both from the RER and SER, what are being synthesized? Proteins and lipids. These are naive proteins and lipids which are accepted by the Golgi bodies. Okay. So from the endoplasmic reticulum to the cis phase of Golgi, the proteins and lipid molecules are being taken. First we will see the function of the Golgi, structure of the Golgi bodies, function of the Golgi bodies, and in which cells Golgi bodies are absent, in which cells Golgi bodies are excessively present. Okay. If you have to look into the structure of Golgi bodies, close towards the nucleus, We have looked into the endoplasmic reticulum. So, this endoplasmic reticulum, we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Cis phase is present towards the nucleus. Cis phase is present towards the nucleus. Fine. Yes. So, these are lamellar structures called as what? Cisternae. Disc shaped lamellar structures called as cisternae. Okay. The face which is facing towards the RER and SER, towards RER and SER, smooth endoplasmic reticulum and rough endoplasmic reticulum. This phase of Golgi is called as cis phase or forming phase or convex phase. Cis or forming or convex phase is towards the nucleus or towards the endoplasmic reticulum. Okay. Trans phase, mature phase or concave phase is towards the plasma membrane. Okay. Cis phase, forming phase or convex phase is towards the nucleus or endoplasmic reticulum. Trans phase, mature phase or concave phase is towards the plasma membrane. So, these lamellar structures are what? Cisternae. Naive proteins and lipids will be accepted by the cis phase and they will get packed inside the Golgi bodies. Packed inside the Golgi bodies. So, inside Golgi bodies packaging of lipids and proteins will happen and this will form out glycolipids and glycoproteins. After packaging is done inside the Golgi bodies, the glycolipids and glycoproteins are formed. Along with this, small vesicular structures which are rich in the hydrolytic enzymes will be synthesized into the cytoplasm and these vesicular structures are called as lysosomes. Lysosomes we will learn detailedly in the next class. So, about glycolipids and glycoproteins, what is the site of the synthesis of glycolipids and glycoproteins? The site of synthesis of this is Golgi bodies, remember that. And we have learned the carbohydrates are present in the form of glycolipids and glycoproteins in the lipid bilayer. So, this glycolipids and glycoproteins which are being synthesized by Golgi bodies are transported to the plasma membrane. Okay. The vesicular structures, glycolipids, glycoproteins will be transported through the trans phase or mature phase to intracellular and extracellular targets. Okay. Now, we will be looking into the presence and absence of Golgi bodies. Golgi bodies are absent in Golgi bodies or Golgi apparatus. This is named after the scientist Camillo Golgi. Fine. They are absent in mature mammalian RBC cell, mature mammalian RBC cell, sieve tube cells and sperm cells of bryophytes and pteridophytes. Those are nothing but anthrozoids of bryophytes and pteridophytes because their mode of transport is what? Water. Mucilaginous sheathing is not required for them. That is the reason in anthrozoids of bryophytes and pteridophytes Golgi bodies are absent. We can simply say sperm cells or anthrozoids of bryophytes and pteridophytes. Clear, right? And then in which type of cells Golgi bodies are excessively present? Excess in root cap cell of plants, root caps. Root cap, which is thimble like structure, positively geotrophing, Golgi bodies which are present in the root cap will give that mucilaginous coating for this root cap and aids in the movement of positive geotrophism. And second cells is acrosome of sperm cells. Okay, in these cells Golgi bodies are excessively present. In the acrosome of sperm cells, Golgi bodies are excessively present. And if you see, Golgi body is unicisternal in yeast. In yeast, Saccharomyces cerevisiae, that is unicellular fungus, Golgi bodies are unicisternal. That means it has single cisternae. And if you see plant cells cisternae are not connected to each other. Okay, cisternae are not connected to each other. So such type of Golgi bodies which are seen in the plant cells are called as dictyosomes. They are called as dictyosomes. And in animal cells, Golgi bodies are profusely branched. This is all about the two organelles that we have discussed, single membrane bound organelles, endoplasmic reticulum and Golgi bodies. In our next class, we will be looking up to lysosomes and vacuoles in detail. Along with that, we will also look into the cytoskeleton. So, that is all for today's class, and thank you.